Hello friends, I'm Dr. Shilin Kumar Singh and in today's class we are going to start about the carbohydrate metabolism. In the last presentation we have seen that our life is highly ordered, means every process from birth to death in the living organism is very systematic and occur timely. But this order of life requires a continuous influx of free energy. ATP is the energy currency and it is the only splendid energy. So how this ATP form from the food we eat? So from today's lecture, we will start a journey to learn ATP formation from the different metabolic pathways. So let's start. So carbohydrates are the most abundant biomolecules on the earth. They are the power nutrients and primarily produced by the plants. Each year, photosynthesis converts more than 100 billion metric tons of CO2 and H2O into cellulose. Carbohydrate metabolism is the whole of the biochemical processes responsible for the metabolic formation, breakdown, and interconversion of carbohydrates in living organisms. So this slide shows the different kinds of carbohydrates. As you can see, carbohydrates are the central to many essential metabolic pathways. So carbohydrates are polyhydroxyaldehydes and ketones, or substances that yield such compounds on hydrolysis. And the empirical formula of this is CH2O power n. And technically they are hydrates of carbons, consist carbon, hydrogen and oxygen, and usually with a hydrogen oxygen hydrogen and this oxygen ratio is 2:1, something like water. We know that carbohydrates is very important part of our energy storage. Actually, two biomolecules, carbohydrate and lipids, both are in energy storage molecules. But carbohydrates are generally available as immediate energy source, whereas lipid act as a long-term energy source and tend to be utilized at the slower rate. And the glucose, the prevalent uncombined or free sugar circulating in the blood of higher animals, is essential to cell function. So the proper regulation of glucose metabolism is of paramount importance to survival. So let's start the journey. As you can see in the slide, the stage one of the carbohydrate metabolism is the digestion of carbohydrates. And this digestion begins with the mouth. When we eat anything, digestion begins in the mouth, where salivary glands, our salivary glands release amylase, which breaks down the polysaccharides, as you can see in this animation. And this polysaccharides converts into the smaller polysaccharide, dextrins and maltoses and some glucose. After mouth digestion, the digested food continues its journey in the small intestine, where pancreatic amylase hydrolyzes these dextrins to maltose and glucose, as you can see in this slide, in this picture. And in the last, these maltose and lactose and sucrose are further hydrolyzed into monosaccharides. And these monosaccharides enter into the bloodstream. So now we know that the food is digested in the mouth and enter into the blood after the digestion of carbohydrate. Now let's move on to the how this produced glucose enters into the cells. As you can see in this animation, after carbohydrate digestion of food in a small intestine, monosaccharide, which is a monosaccharide glucose, which is a six membrane ring molecule, it enters through a specific transporter proteins, and it moves from outside cell into the cell cytosol, and this cytosol is the site of glycolysis. All of the glycolytic enzymes are found in the cytosol, and cellular freight of the glucose begin with glucose transport and phosphorylation. And there are so many pathways in which this glucose is utilized: anaerobic, aerobic, glycolysis, glycogen formation, and conversion of other intermediates. We will discuss with these things in our later classes. Now let's move on to the next slide and see the major pathways of the carbohydrate metabolism. As you can see, the glucose is the major fuel of the most organism, and the major pathways of the carbohydrate metabolism either begin or end with glucose. It starts with the process known as glycolysis, and then the process goes like the modification of pyruvate into acetyl coenzyme A, and this it is done by pyruvate dehydrogenase complex. And then the third stage of the process is known as Krebs cycle, which you can see in this slide also. And the fourth stage is the process known as electron transport chain, in which the energy is generated. So the carbohydrate metabolism in metabolism can be classified into these six these six pathways. Glycolysis is the oxidation of glucose into pyruvate, and citric acid cycle after oxidation of pyruvate to acetyl coenzyme A, and hexose monophosphatation. It's a we can enable cells to produce ribose 5-phosphate and NADPH. Then glycogenesis, synthesis of glycogen from glucose, glycogenolysis and glyconeogenesis. These are the different pathways we will discuss in the later classes. Now let's move on to the word. Glycolysis word is derived from the Greek word sweet, Greek like and lysis, sweet and splitting. It is the metabolic pathway that uses glucose, a digestion product, and convert them into pyruvate. pyruvate. Glycolysis occur in all human cells. In RBCs, glycolysis is the major source of ATP because RBC lack mitochondrial oxidation. So glycolysis is believed to be among the oldest of all of the biochemical processes. It is the only pathway that can produce ATP in the absence of O2, oxygen. Actually, this metabolic process serves both aerobic and anaerobic cellular respiration. Aerobic means 6 carbon glucose converted into 3 carbon pyruvate molecules, while anaerobic means no oxygen in the no oxygen conditions. In these conditions, glucose converts into the lactate. Now let's move on to the next slide and see. This is the different step. These are the different steps of the glycolysis. As you can see, uh, glucose which had been derived from food after digestion, it's converted into the pyruvate through a series of biochemical reactions, and we will see step by step of this pathway into the later slides. So if we can divide 10 reactions of glycolysis into 3 stages which occur in the cellular cytoplasm. The glycolytic pathway first stage is priming stage, in which glucose is trapped in the cell and destabilized. And second stage is splitting stage. Two interconvertible 3 carbon molecules are generated and the cleavage of 6 carbon fructose. And the third stage is oxidoreduction phosphorylation stage, in which ATP is generated. We will discuss all these steps in incoming slides. These are the first stage of glycolysis, uh, which involves energy investment of 2 ATP. It is from reaction 1 to 5. Glucose is first converted into the first 1,6-phosphate, and the series of steps use up to 2 ATP. We will see in this next slides. So this is the overall glycolysis pathway. You can see glucose which had been derived from the food after digestion, it's converted into the pyruvate through a series of biochemical reactions. We will go through these reactions step by step in the later slides. So this is the most important step of the glycolysis. In this step, the glucose ring is phosphorylated, which means addition of phosphate group from ATP molecule. As a result, at this point, glycolysis, one molecule of ATP has been consumed. And important thing is that it is irreversible in nature. Actually, after entering the glucose into the cell, phosphorylation takes place. This phosphorylation prevents the transport of glucose outside the cell and increase the reactivity of oxygen in the resulting phosphate ester. So the commitment for this step dictates the commitment for the cell to continue the glycolysis process. So this is a determining step. And one more important thing is that atomic magnesium is also involved to help shield the negative charges from the phosphate group on the ATP molecule. Now let's move on to the next slide, next step. And this is the second step, glucose 6-phosphate to fructose. In this second step, glycolysis, glucose 6-phosphate phosphate is converted in the fructose 6-phosphate with the help of enzyme phosphoglucoisomerase. It involves a isomerization reaction, as you can see in the animation. As the name of the enzyme suggests, this reaction involves an isomerization reaction. Reaction involves the rearrangement of carbon-oxygen bond and transform them into 6 carbon ring into a 5 membrane ring. So this kind of rearrangement takes place. Now let's move on to the next step. Now the step is the fructose 6-phosphate to fructose 1,6-bisphosphate. In this step, with the help of phosphofructokinase and magnesium as a cofactor, fructose 6-phosphate changes into the fructose 1,6-bisphosphate. Similar to the reaction that occurs in step 1 of the glycolysis, a second molecule of ATP provides the phosphate group that is added onto the F6P molecule, fructose 6-phosphate molecule. Okay, now let's move. And after this step, the next step is the fructose 1,6-bisphosphate, which is the 6 carbon molecule. Now break down into the 3 carbon molecules, 2 3 carbon molecules. So this is the splitting stage. So this is the splitting stage in which fructose 1,6-bisphosphate breaks down into two molecules, DHAP and GAP, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate is very important molecule because it serves as a substrate for the next reaction in the glycolysis. Now let's move on and see. Now the energy investment reactions of the glycolysis are over. Let's move on to the energy production reactions of the glycolysis, in which 4 ATP energy molecule is generated. So now we are at this reaction, at the reaction 5 of the glycolysis. Both are DHAP and G3P, both are interconvertible. You can see that interconversion of GAP and DHAP. At this point in the glycolytic pathway, we have 2 3 carbon molecules, but have not yet fully converted into glucose into pyruvate. So prevent the loss of other 3 carbon unit, triose phosphate isomerase catalyzes the interconversion of DHAP and GAP. Because of this reaction, the original molecule of the glucose has been converted into 2 molecules of GAP. Now let's move on to the next reaction, the glycolysis. In the step 6 of the glycolysis, two main events take place. First is glyceraldehyde 3-phosphate is oxidized by the coenzyme nicotinamide adenine dinucleotide NAD, and the molecule is phosphorylated by the addition of a free phosphate group. So the enzyme that catalyzes this reaction is the glyceraldehyde 3-phosphate. This enzyme is a tetramer, means it has 4 arms or subunits, and each subunits contain one binding site for GAP and another binding site for NAD+. On to the seventh reaction of the glycolysis, which is a oxidoreduction phosphorylation stage. In this step, 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate with the help of enzyme phosphoglycerate kinase. This reaction involves the loss of phosphate group from the starting material. So the phosphate is transferred to a molecule of ADP and we yields our first molecule of ATP. So We actually have two molecules of 1,3-bisphosphoglycerate because you can remember that two 3-carbon products from the stage 1 of glycolysis. So we actually synthesize two molecules of ATP at this step. So with this synthesis of ATP, we have cancelled the first two molecules of ATP that we used and leaving us with a net zero ATP molecules up to this stage of glycolysis. Now let's move on to the next eighth reaction of the glycolysis, and this reaction is 3 Pg to 2 Pg, 3-phosphoglycerate to 2-phosphoglycerate. In this step, a simple rearrangement of the position of the phosphate group on the 3-phosphoglycerate molecules, making it 2-phosphoglycerate molecule, using a phosphoglycerate mutase. This mutase enzyme that catalyzes the transfer of functional group from one position on a molecule to another. So this is simply rearrangement reaction. Now let's move on to the next reaction, and this is reaction 9 of the glycolysis. And enzyme enolase removes a molecule of water from 2-phosphoglycerate, and we form phosphoenolic pyruvic acid (PEP). Now let's move on to the 10th reaction of the glycolysis, which is a final reaction. So finally, enzyme pyruvate kinase transfer a phosphate from phosphoenol pyruvate to ADP and to form pyruvic acid and a 1 ATP in the step 10. So let's take a quick review of the all of the reactions. Suppose this is our cell, and in this cell, this part is the cytosol. And as you can see, the glycolysis occur glucose to pyruvate with the help of 10 steps. So in these two steps, ATP is formed, and this one, sixth one is the ATP indication. So now we can divide. See, in this animation, glucose is going into the pyruvate. This is the first stage. The enzyme hexokinase phosphorylase and adds phosphate group to glucose in the cell cytoplasm. And this phosphate, now let's move on to the next step, and with this enzyme phosphoglucomutase, and it isomerizes glucose 6 phosphate to fructose 6 phosphate, and this fructose 6 phosphate is now form fructose 1,6 bisphosphate with the help of phosphofructokinase enzyme. Okay, now this triose phosphate isomerase rapidly converts this fructose bisphosphate aldolose, break down the fructose 1,6 bisphosphate into DHAP, three molecules, and these three molecules of DHAP undergoes into the fifth stage with the help of triose phosphate isomerase, and this triose phosphoisomerase it's convert into the step 6. So this we have seen that energy consumption phase, and now the energy production phase. These two molecules of GAP, glycerol 3 phosphate, is going into the stage 6, in which a phosphate group is adding gap to form 1,3 is phosphoglycerate. So this glycerol 3 converted into 1,3 bisphosphate. Now this is the step 7, and you can see DPG is converted into the 3-phosphoglycerate PGA with the help of phosphoglycerokinase. And now the 8th step. In this step, you can see 3 PGA, 3 PGA molecules from the third to second carbon form two phosphoglycerate molecules. And this is the 9th step, in which enzyme enolase removes a molecule of water from 2-phosphoglycerate and form phosphoenol pyruvate. This happens for each molecule of 2-PGA from step 8. And this is the final stage. Enzyme pyruvate kinase transfer a phosphate group from phosphoenol pyruvate to ADP and form pyruvate and ATP. So this happens for each molecule of PEP, and this reaction yields two molecules of pyruvate. So from this you can see that glucose to pyruvate. Now let's move on to the next slide and see how much ATP is produced, overall ATP production. Glycolysis 2 ATP add phosphate to glucose and fructose 6 phosphate. So 2 ATP is consumed and 4 ATP are formed in the energy generation by direct transfer of phosphate group to 4 ADP. So there is a net gain of 2 ATP and 2 NADH as you can see in the table. Now let's move on to the regulation of glycolysis. Glycolysis is regulated by three enzymes, reaction 1, 3 and 10. Hexokinase, phosphofructokinase and pyruvate kinase. And besides three enzymes, hormones also regulate glycolysis. For example, glucagon inhibits glycolysis by repressing the synthesis of fructose 2,6 diphosphate, while insulin promotes the glycolysis by stimulating the synthesis of fructose 2,6 diphosphate. And this is with this memorizing trick, I am ending this lecture, and you can remember these steps with the help of this quotation. Goodness gracious Father Franklin did go by picking pumpkins to prepare pies. So these are the simple trick to remember all of the steps of the glycolysis. And so these are the questions which you have to answer after going through this presentation. What is glycolysis cycle? How many steps of the glycolysis? So you can answer all of these questions onto my email ID drshair82@gmail.com.